In this video, we will discuss about transposons. Transposons are also called as transposable elements. Transposons are also called as jumping genes, mobile elements, cassettes, insertion sequence. This term was given by scientists Hedges and Jacob in 1974. So we can say transposon are class of genetic elements that can jump to different locations within a genome. Now we'll explain. This is transposon, also called as IS mean insertion sequence. Now this is the DNA where this will get integrated. Supposing at this place, this insertion sequence is being integrating into this DNA genome, and from here on the lower strand on this strand at this place. So we can see when this will get inserted into the DNA, we can see the yellow sequence. Now TG will remain here, A, C, C, G, T, C, G, G, A. Here it will be TG at this place and yellow sequence again on this side. So at this place, this insertion sequence or DNA has been integrated into the DNA of another genome. This transposon is insertion sequence of one DNA, and this yellow colored DNA is DNA of another genome. So they are getting integrated with each other. Now this red color sequence of bases have been shown. This has been completed with the help of DNA polymerase that will add bases opposite to this, and this sequence will be completed. So this targeted gene has been disturbed by the insertion of this sequence. So naturally, codons will be different, now protein will be different, and this will also change the genetic expression also. So mutation may occur. So this is how insertion sequence get integrated into the other genome. Now next is, this is transposon. I'm going to explain about inverted repeats in this sequence. Like this is the transposon. This part is called as inverted repeat. A, B, C, D, E. Now here we can see on the opposite strand, this will be A, B, C, D, E. So this is called as inverted repeat. Means same sequence is being repeated, but this is in a inverted manner. So this is called as inverted repeats. Now here opposite to A will be its complementary sequence. So this sequence is complementary to A, B, C, D, and this sequence is complementary to A, B, C, D, E. This is hypothetically written A, B, C, D. Otherwise, these will be the nitrogenous bases present in the DNA, like we have seen here. Opposite to A will be T, opposite to C will be G, then opposite to G will be C. Now, we can define transposable elements. Transposable elements can be defined as small mobile DNA sequences that move around chromosome with no regard for homology, and insertion of these elements may produce deletion, inversion, and chromosomal fusions. Like if we have this DNA and this is the another DNA, supposing it's this part is getting integrated into this DNA here, this copy is being integrated here in this DNA. Now when this is getting integrated, they are not homologous to each other. This is a different genome, this is different genome. So there is no homology, there is no similarity between them. Now when this DNA is getting integrated from here into this, so this is causing additions of the genes, right? So certain changes may occur. Genes may get deleted here at this place, they may get deleted, they may show inversion, they may show different kind of chromosomal aberrations. Now this phenomena was observed by scientist Barbara McClintock. Barbara McClintock is scientist who was studying genetic instability in maize plant in 1965. Now this is Barbara McClintock. She was doing a lot of work on the maize plant, and she observed transposon in maize plant. Now these different colored grains present in the maize grain are due to the formation of transposons. And this flower is also showing various kind of variegations showing the transposons. Now she was studying maize grains. Now in maize plant, if we see the general phenomena of fertilization in flowering plant, we will see that there is always a double fertilization. Now pollen tube will come here and this will carry the male gamete. This pollen tube it's carrying two male gametes. So these are two male gametes. Now these two male gametes will enter here. One will fuse with the egg and second male gamete will come and fuse here. And in the center here it is secondary nucleus which is diploid in genetic constitution. And this secondary nucleus which is diploid, and this is the male gamete which is fusing with it. Now three will fuse with each other. Now this will become triploid in genetic constitution, like two nuclei plus one male nucleus. Now this phenomena of one male gamete fusing with the egg is called as and second with the secondary nucleus is called as double fertilization. But we are talking about this part. Here three nuclei are there, triploid condition is there, right? So we can see here in the chromosomes. Now this is the female gametophyte and this is the male gamete, like male gamete which is coming from here. Now this is the chromosome of the female gametophyte having CC gene. So its genome is CC. Now here we will name it as C prime and DS factor is also associated with it. And this is located on chromosome number 9. Now what happens when this fertilization has taken place like this, we have shown here. Now we will get triploid endosperm, like two chromosome of female gametophyte and one of the male. So we'll get three, 1, 2 and 3. This is called as fertilization and this is producing triploid endosperm. Now its genome will become C, C and C prime. Now after some time we'll see that a fragment of this chromosome has been broken up and this fragment will be lost. So this is acentric fragment. Now this will be lost and this is called as acentric, means this is without centromere. So this is called as acentric fragment because it doesn't have any centromere. So this will be lost. So we will be left with CC genotype. This is C, C and this part of the chromosome. So we will be left with genotype CC. Now this C prime gene, this is an inhibitor of the aileron layer of the maize, right? What is inhibitor? I mean, this will not allow the maize gene to show its expression of the color. Now for activation of the C prime, we need to have a gene called as activator. For its breakage from this gene, we need to have DS, which is called as dissociation factor. This is called as dissociation factor, which is causing breakage of the C prime gene. AC is activator. This is activating the dissociation process. Now we'll understand here, this is the maize grain. This part of the maize grain, I've cut its LS of the maize grain. This part is endosperm. Usually grains are yellow in color. This is cotyledon and this part is embryo. But we are talking about this endosperm which is being produced by double fertilization and this is having triploid endosperm. Now this should have been yellow in color but this becomes colored like this. Now we can see here this is showing different colors like purple, red, and this coloration is due to the loss of this gene, C prime gene. Because this C prime gene would have inhibited color formation because this is inhibitor of the color pigmented kernels of the maize grain. But when this is dissociated, this has been lost, then its endosperm will be colored in nature. This become purple, this may red. So this is this AC and DS factor which are responsible for loss of this fragment. They are called as AC and DS are called as transposons. They can get integrated anywhere in the genome, so they are called as transposons, and due to which we are getting the colored grains in the maize plant. Now we'll discuss about characteristics of these transposons. Now transposons, they are DNA sequences which are coding for enzymes whose identical copies inserted into a new DNA site. Now I made this one DNA with the red color, another DNA with the green color. Now this DNA will either replicate or it can break also. This will get integrated into this. Like this part of the DNA has been getting integrated into another DNA. So this is the new DNA where this is getting integrated, right? And its exact copy, one this may get replicated and its exact copy may get integrated over here. When this transposition, this shifting of this DNA segment here, this is also called as transposition, because this gene is now combining with this green DNA. So this is leading to recombination. When its copies are being made, this is called as replication process. The insertion of transposable element disrupts the integrity of the target gene. Now this green DNA was showing certain sequence of the gene like A, T, C, G, C, can be any of the sequence. Now, but when this is getting disturbed by the red genes, now this can be like C, G, A, T, C, G, it can be any sequence. Like this sequence has been disturbed. So when this sequence has been disturbed, its integrity of the target gene mean target gene in which it has been getting inserted, now this will be disturbed, its expression will be different. Now transposon, they carry signals for the initiation of the RNA synthesis. Now this gene, which is of the transposon, this can form RNA, this can form protein, this is carrying the signal. Sometime what happens, supposing this part of the DNA was not active, this was dormant. When this red gene has come into this and get integrated over here, this may activate these dormant genes. So this carries the signal for the initiation of the RNA synthesis. A transposable element is not a replicon. Replicon means like here, we can see here again, this genome where it is found in the red DNA. Now this can replicate because it can replicate with the DNA of its original type. But when this is going to get attached to another DNA, now this cannot replicate. Like phages and viruses, they can replicate, but this DNA, which is mobile DNA, this will not replicate. This will not carry the replicon point. No homology exists between the transposon and the target site for its insertion. Like we have seen, they are not homologous to each other. It is a random phenomena. Any of the DNA can get integrated into any of the genome. So this is not a planned phenomena. This is a random phenomena. So these are different characteristics of transposons. Now we'll discuss about its types of transposons. As we know, we have used the term insertion sequences, mean they get inserted anywhere in the genome, so they are called as insertion sequences. So types, simple transposon or insertion sequences. We can see here, this part is showing IR region, this is also IR region. IR is inverted repeats. This is inverted repeat region. Like we have earlier seen how this is inverted repeat. We have seen in the first slide. Now this part is coding region. Transposase gene, and this can form the enzyme transposase. This transposase gene is responsible for its transposition, mean its insertion into the another DNA. Usually this is made up of 800 to 1500 base pairs, not coding for any of the protein. In bacteria, Escherichia coli, various kind of insertion sequences are found, IS1, IS2, IS3, IS4. Recently in a bacteria, IS21 has been discovered. This is also found in bacteriophages and bacteria. Now, second type of transposons are complex transposons or TN transposon. We can see here, we can see this is one insertion sequence. This is another insertion sequence. Now, they have been combined with the help of a toxin gene. This will also act as a marker gene. So, this is inverted repeat, this is inverted repeat, inverted repeat, inverted repeat, right? So, this is being integrated with the help of toxin gene. And this is made up of inverted repeat region. This region is made up of 8 to 30 base pair. So, this is called as complex transposon, which is getting by combination of two or more insertion sequences. So, this is called as complex transposon. Now, we'll discuss about its examples of transposon. First, we'll discuss about TN3 transposon of Escherichia coli. Now, this is made up of three genes, TRPA, trip P and BLA, and 38 base pair part is inverted terminal repeat. This is also 38 base pair inverted terminal repeat. Now total sequence of the genome, that is of transposon, is made up of 4957 base pairs. Now trip A will form an protein called as transposase. This is made up of 1015 amino acids. Trip B form protein called as or enzyme resolvase repressor. This is made up of 185 amino acid. Now this resolvase or repressor will help in regulation of the transposase. Transposase is responsible for its transposition, its integration into the another genome. Now BLA, beta lactamase, and this gene is responsible for conferring the resistance to the antibiotic, that is ampicillin. Like what is the advantage of an antibiotic resistant gene? We are able to identify the genome. This is acting as a marker gene, and this will also provide resistance in presence of antibiotic. We can grow the culture in presence of antibiotic ampicillin. Now second type are bacteriophage mu. Mu word is mutator. For this again we are taking the example of Escherichia coli genome. This is lac plus gene. Lac means this is helping in utilization of lactose. Like when we add the lactose into the media, lac plus gene will help in utilization of the lactose. Now what has happened? Into this genome, this sequence has been inserted. Now this sequence is from the bacteriophage. Now what are bacteriophages? Supposing this is bacteria, and we will see here, this is a virus which is parasitizing it. This is its DNA, and this is the DNA of the bacteria. So this is the genome of the bacteriophage. This genome has been getting inserted into the bacterial DNA. Like supposing this is the bacterial DNA, and this gene is getting inserted over here, right? So this lactose gene will get disturbed. Now this will not work for the lactose utilization. Now this has caused the mutation because genes will change. So this is called as bacteriophage mu type. So this is also called as temperate bacteriophage, as well as giant transposon. This part is called as giant transposon. So lactose gene will not work because of integration of the mu DNA of the viral particle. That is, this virus will send the DNA into this and they will get integrated. So this is called as transposable elements. Now next is yeast TY element. Now this is found in yeast. As we know, this is yeast is a single celled fungus having nucleus. Now here we will see that yeast TY elements, they are made up of TYA and TYB, and they are surrounded by delta sequences, now which is made up of 340 base pairs. Now about yeast TY element has 35 copies. This is made up of 5900 base pairs, and they have long terminal repeats. It's not inverted terminal repeat, they have long terminal repeats. Now here we can say this is made up of delta sequence, which is of 340 base pairs, and these are also found as retroposons. So total length is 5900 base pairs. Now this is about types of the transposon. Now what is its significance? Now when one DNA getting integrated into another DNA, now this will increase the genetic diversity. Genetic variation will improve. This also plays a very important role in the evolution process and also helps in gene regulation in all the organisms which are carrying the transposons. Transposon also cause mutation because wherever they will get integrated, they will change the DNA sequence, they will change the codon, they will change the protein, they will change the gene expression, they will change the gene regulation, and ultimately they will lead to the mutation. And due to mutation, there is evolutionary process, and they are also causing the evolution process. This also alter the gene expression. As we know, when mutation occurs, gene will not express like the original way, its expression will change, and ultimately genes will get modified, they will get diversified, and these transposons are also responsible for various kinds of abnormalities. And sometimes when they are getting integrated into the dormant genes are genes which are not working, which are not expressing themselves, but if transposon is carrying its RNA initiation signal, this can activate those dormant gene for its functioning. So this is about significance of transposon. So in this video we have discussed what are transposon, what are its types, and what is its significance. If you enjoyed my video, please like, share and subscribe. Thank you for watching.